Hello and welcome to my presentation. My name is Jasjit Jolly and I will be presenting on secondary metabolites and bees. Now, my main takeaway before I get into that, I think it's important to understand my question. My question being, are visitation rates made by bees to flowers affected by certain secondary metabolites that are located in the pollen of the observed flowers? In this project, secondary metabolites being compounds that aid flowers in basic survival, with things such as detection and stuff like that. Now, my main finding is after conducting an ANOVA test, we found that they are affected by secondary metabolites. When it comes to my methods, I pulled my first paper from Dryad. It was a data set titled Bees Use the Taste of Pollen to Determine Which Flowers to Visit. Basically, what I did was from this data set, I found a column of data that basically, with the variable name, number of feeding bouts. Now you don't see the x-axis, but basically what's supposed to be on the left side of this table is which treatment the plant is being given. So which secondary metabolites are we looking at, and how many times does a bee go to a plant with a certain treatment, a certain secondary metabolite. And basically, I used points from this column to conduct an ANOVA analysis. Now, I was lucky enough that my second paper had a very similar variable, so I did not have to conduct any sort of variable comparison calculation. This paper comes from, again, from Dryad. It's titled Specialized Metabolites and Floral Resources, Effects and Detection in Buff-tailed Bumblebees. And again, the table is very similar. You don't see the x-axis, but it does show which treatment, which plant is being given. So which secondary metabolite with a certain plant, how many times are the bees going to that plant with a specific secondary metabolite. And again, very similar to the last paper, I used points from this column to conduct an ANOVA analysis. So these two data sets put together, I did an ANOVA analysis. The results from this ANOVA analysis are as follows, starting off with some very basic summary statistics. There are 6 columns representing 6 different secondary metabolite treatments with things such as the count, sum, average, and variance. I calculated standard error on my own for error calculations, but the main meat of the ANOVA analysis is the F statistic and the F critical value. It found that with the F critical value of 2.24, the F statistic was actually 13.2. Which is much, much greater than the F critical value, which for an ANOVA analysis that uses categorical data, this means that we can reject our null hypothesis. And with a setup like this, our null hypothesis is basically looking to see that between each of these 6 different columns, these 6 different samples with different secondary metabolites, are the means of each of these samples equal or are they not? The null hypothesis says that they are equal. We reject this, and we can conclude that they are in fact not all equal, given the F statistic, comparing it to the F critical value. Now, here is a figure from my ANOVA analysis. It has a lot of the summary statistics that you saw on the previous slide, with some error bars representing the standard error. And it's plotted on a graph with the x-axis representing secondary metabolite type, and the y-axis representing average number of times bees visited the flower. While it's not, you can't fully say which specific treatment, sort of had the best effect on bees visiting them, because the main question, again, was looking to see if secondary metabolites even matter. Nonetheless, from this graph, we do see that cellulose has the highest by a very, very small margin over sucrose. But before that was even done, I looked at each and every single treatment, and I looked at the data that it had just to see if there's any outliers. So stuff like skewness, range, and other summary statistics were looked at. Who cares? The rationale. The majority of flowering plants require that insects pollinate them in order to be fertilized and create the next generation of plants. I looked at bees so I can better understand what exactly allows for certain flowering plants to thrive and pass on to the next generation versus those who fail to accomplish this. The ANOVA analysis compared the secondary metabolites found in these plants to the success of attracting pollinators. This will shed light on whether or not secondary metabolites even have such a role in predicting the success of bees pollinating flowers. Thank you for listening to my presentation.